Warahmatullahi wabarakatuh. This is the slide regarding to the nucleic acid and the genetics. So I would like to give a talk about this. So we're going to start with the first slide. So in this chapter or in this subtopic, so the student must, at the end of the course, the student must know the chemistry of the nucleic acids, and then the student can describe the structures of nucleic acids and the chromosomes, and the student must also explain how the quality and the information genetic materials are conserved in generation after generation, meaning that how the DNA can be transferred from the mother cells to the daughter cells. All right. Okay. And the last one is the student should know the different types of RNA. For example, the rRNA, the tRNA, mRNA, and hnRNA. All right. So we move on to the next slide. So this is the topic in a glance. So the student will start with the nucleotide, learning about the nucleotide, and from the nucleotide, how it becomes genes, and then after the gene, it involve the histone and the nucleosome, and then lastly, it will form the chromosomes. All right, and then so we go to the chemistry of the nucleic acid. Okay, so there are two types of nucleic acid. So the first one is the DNA, stand for the deoxyribonucleic acid, and the other one is RNA, which is known as a ribonucleic acid. All right. So the DNA is a more stable as compared to the RNA because the DNA is double-stranded, RNA is single-stranded. All right. So the function of the DNA is to store the genetic blueprints of the cells, meaning that they store all the information about the genetics. But the RNA, it's a form that can transfer the genetic into a protein, for example. Okay, so it has the code to be decoded and to be translated into the protein. Okay, next move on. So the nucleotide is the basic unit of the DNA. All right. So it consists of the phosphate group, the pentose sugar, and the nitrogenous base. All right. So the nitrogenous base can be divided into two different types, which is the purine and pyrimidine. So the purine has two carbon ring, but the pyrimidine only have one carbon ring. So the purine, the purine nitrogenous base is adenine and guanine. For the pyrimidine is cytosine, thymine, and uracil. So for the thymine, it only can be found in the DNA. However, the uracil is only found in the RNA. All right. So you must know there's two types of things that you must know here. So the first one is nucleoside, and the other one is nucleotide. So the nucleoside consists of nitrogenous base and pentose sugar. But when they include the phosphate group, it the name changed to the nucleotide. Okay, meaning that the nucleotide must have the nitrogenous base, pentose sugar, and the phosphate group. All right. Okay, let's move on. Okay, so in this nucleotide, there's a bond known as a phosphodiester bond. So the phosphodiester bond bind to the carbon number 5 of the pentose sugar. The other phosphate group will bind to the carbon number 3 on the pentose sugar. So this is the phosphodiester bond that makes the strand of the DNA and RNA. Alright, so we move on to the next slide. So like I said before, the phosphodiester bond can build the nucleic acid chain. Okay, so the nucleotides in the nucleic acid strand are linked together by phosphodiester bond. Okay, the phosphodiester bond is formed between the 3' hydroxy group, meaning that 3' of hydroxy group of the pentose sugar, meaning the carbon number 3, and then the 5' phosphate group of sugar, the pentose sugar of the next nucleotide, meaning that the first nucleotide and second nucleotide, they bind to the different carbon. Alright, okay, so the directionality of the chain is the 5' end with the phosphate group attached to the 5 carbon, and the other one is 3' end, which the hydroxy group attached to the 3 carbon, the carbon number 3, the third carbon. Alright, so we move on to the next slide. So from there, when it involves many numbers of nucleotide, So it will become a gene. Okay, so nucleotide number 1, 2, 3, and 4. Like I said before, the nucleotide must have three, consists of three, which is a phosphate group, the pentose sugar, and then and the base. Okay. In this figure, you can see that the phosphate backbone is very important. Okay, so it makes the backbone of the strand. Okay, so from here you can see, from the left side you can see this is the other one, this is one strand, then the other one is on the right side, the second strand. Okay, so the nucleic acid sequence is represented by the sequence of nitrogenous base. Like I said, the nitrogenous base has a thymine, TACG, okay, TACG, and by convention, the sequence of nitrogenous base is written in the 5' to the 3' direction, and there's a base pairing here. So the base pairing is T will be paired with the A, C will be paired with the G. All right, okay. So you can see here there's a two strand of the DNA, and then it bond together in the middle of it with the hydrogen bonds. So now we go to the next slide. Okay? All right, so that's the hydrogen bonds. As you can see from this image, there is two strand here. The first strand is in this blue color. There's a thymine nucleotide, and the other one is guanine, and the other one is adenine and cytosine. All right, so from here you can see that the hydrogen bonds can be found when the H from the adenine bond bind to the O atom. Okay, so it will create, or it will develop hydrogen bonds. So you must know that the hydrogen bonds is weaker than an ionic bond or covalent bond. Okay, but it's stronger than the van der Waals forces. Okay, so you have to bear in mind that the hydrogen bond donors are only those 4H atoms which bound to the electronegative atom, such as N or O. Okay, from here you can see that O, it binds to the O, and this hydrogen atom bind to the N. Okay, this is very interesting, whereby the TA will have a hydrogen bond with two hydrogen bonds, as compared to G to C. There's a three hydrogen bonds, which is more hydrogen bonds, is much stronger. All right. Okay, so we go to the next slide. It's regarding to the structures of nucleic acid and the chromosomes. All right, so this is the different levels of chromosomal organization. So it's just to give you an idea for the whole thing that you will learn. All right. Okay. So you you start with the DNA double helix. So and then you move to the nucleosome, whereby the nucleosome will involve the central histone and the DNA. And then from the nucleosome it will develop into the chromatin fiber, and then the chromatin fiber will coil and make a supercoil within chromosome, and lastly there will be a chromosome. All right. Okay, so we start with the DNA history. So the DNA history started at 1928, okay, by the Frederick Griffith, and then after that, followed by the Oswald Avery, Colin McLeod, and Maclean McCarthy. Okay, so they conduct an experiment regarding to they work on the different bacteria, which is different strain of bacteria, the S strain, which is a smooth, because the bacteria have some protein coated outside of the bacteria, so it to make it smooth, and the other one is rough bacteria without coated. Okay, these two bacteria were transfected to the mice, and then they found that the virulent bacteria is the one that have the coated one, the protein coated one. Okay, so they found that when transfected to the mice, they kill the mice. Okay, and then they try to kill the bacteria with the coated one, and then they try to culture culture together with the rough one, and then they transfect to the bacteria. So transfect to the mouse, and then the mouse die. Right. Alright, so they found that, meaning that the virulent or the heat kill virulent bacteria has transferred the DNA to the bacteria which is to the to the rough bacteria. So meaning that there's a genetic materials that can be transferred into other organisms that can be delivered to the host. All right. Okay. So after that, moving on to the to 1952, the two scientists, Alfred Hershey and the Martha Chase. So they confirmed about the DNA, and then it's about an experiment. I think they did some coloring. I mean, not dye. They use a virus. Okay, they use the virus to transfect the bacteria. Okay, so they use the virus. So the virus, as you know, that the virus must have the RNA, and then they have a protein coating outside there. All right, so they use a labeling chemical to label the protein in red color, and the other one they use a green color to label the RNA. And then after that they did transfect the bacteriophage in so they found that only the green color has been found in the bacteria, not the red color, meaning that the red color representing the protein, there's no protein inside that. There's only the green color, meaning that the DNA only has been transferred into the bacteria, but not the protein. So they conclude that the DNA is the genetic material that can be transferred into other organisms. All right, and then moving on to the 1953, James Watson and Francis Crick has won the Nobel Prize by developing or by setting up structures of the DNA double helix. They introduced the structures of the DNA double helix. So this is quite history. All right, so we move to the DNA double helix. All right, so DNA double helix. The important thing that you must know about the double helix is there's a two different strand, okay, which bound together in the middle of it with the hydrogen bond. Okay, so most RNA consists of single strand of polynucleotide, and most DNA exists as a double-stranded chain which is twisting together forming a double helix. But there's a section for the virus called parvovirus because they only have a single-stranded DNA. Okay, move on is what is the characteristic of the DNA double helix? So both strands are antiparallel, okay, which is run in opposite direction. From the left side, it runs from the top to the bottom, 5 prime to 3 prime, and the other one from the bottom to the top, the same direction. I mean the same prime, 5 prime to 3 prime. Okay, then there's a sugar phosphate backbone found on the outside, and in the inside one there's a nitrogenous basis. Okay, so this is very important for the DNA to the nitrogenous base must in the inside is very important because it can conserve the genetic material. Okay, and most importantly is the DNA charge. So DNA is negatively charged, so this is because of the phosphate group on the DNA. Okay, so the other characteristic of the DNA double helix is that the nitrogenous bases are stuck inside and protected from damage by environment. That's the important thing, why the nitrogenous base must be inside. Okay, so the other one is the different spaces exist. So if you can see in here, there's a minor groove and a major groove. So these groove will allow the binding for proteins that involve DNA synthesis, meaning that it's involved during the replication of the RNA synthesis or during the transcription. Okay. Okay. Okay, so the next the characteristic of the DNA double helix on the base pairing. So the base pairing, the bases of the one DNA strand are paired with the bases of another strand by hydrogen bonds. Like from the image here, from just on your side, on your right, there's a two DNA strand, and in the middle you can see the nitrogenous bases are connected to each other. So it connected by the hydrogen bond. All right. So the adenine pairs only with the thymine with two hydrogen bonds. The other one is guanine pairs only with cytosine by three hydrogen bonds. So this is the important thing. Important thing to know that the T must bind and must bond with the adenine. The cytosine must bond with the guanine. Okay. These two strands are said to be complementary to each other. All right, so I have one exercise here. So if sequence of one change is given in this box, so the sequence of opposite strand can be determined. Okay, like from here we can see that in 5 prime it start with the A, so A must complement with the T. Okay, so the T must complement with the A, and so on and so forth. All right, so this is the answer. So the sequence is for 3 prime to 5 prime is T A C G C G A T C G A G T A A A. Okay, the other characteristic that you have to know is that the DNA double helix can be denatured. So it's called the process is called denaturation. So it depends on two different parameters. So the first one is pH. If the pH is more than 10, meaning that it's an alkaline pH, so this pH can deprotonate bases of the DNA, and this can destroy the base pairing. Okay, so it will separate the double helix into the two single strands. But however, the alkaline also can cleave phosphodiester bond, but only if in RNA. So the second one is the temperature. All right. So for the temperature, heating can separate into single strand. Okay. So there's two types of temperature that you have to know. First one is a melting temperature, and the second one is the re-annealing temperature. Okay. So the melting temperature is considered is when the 50% of the strand has been cleaved or has been separated. And for the re-annealing temperature is the temperature at the lower temperature where the double helix can hold together again. All right. So another important thing about their DNA double helix is about their conformation. So they have three alternative conformation. So the first one is ADNA. So the ADNA is a right-handed conformation, and it's produced by the moderate dehydration of the B form. So it's actually from the B form. Because of dehydration it becomes a ADNA. So for example, the DNA and RNA hybrid, and the RNA double-stranded region, and so on and so forth. Okay, the second conformation is the bDNA. So the bDNA also is a right-handed. So examples of the bDNA is the chromosomal DNA. So this conformation has been proposed by James Watson and Francis Crick, where they won the Nobel Prize. They determined the structures of the DNA units in 1953. All right, the last conformation is the zDNA. So the zDNA is quite different. So it's a left-handed and it has a zigzag shape. So so this is has been used. This is has been proposed by other scientists. Okay, so next is the DNA double helix in supercoiling. So DNA is coiled and in the form of double helix. So further coiling of the double helix can produce a supercoil, meaning that the DNA coil become a twisting. So it will have a twisting structures. So this supercoiling facilitates the compaction of the DNA in the bacteria or in the viral. Okay, so there are also the linear DNA. This is occurs during DNA and RNA synthesis. This is especially for the replication and for the transcription. All right, guys. So there's another place or another location that DNA can be found. So in this slides, I would like to show that the DNA can be found in prokaryotes. The DNA can be also can be found in two different types, which is the plasmid DNA and the chromosomal DNA. So that's for prokaryote. Then in the eukaryote, the DNA also can be found in the mitochondria. So in the mitochondria, the DNA is in circular conformation. Okay, so this slide shows the difference between the prokaryote and eukaryote in terms of the DNA type, the structure, the location, the DNA packaging, the function, and gene copy of number. So I think this slide, I think you can read on your own, because I have put all the things, all the important things in here to make it simple for you to study. Okay, now we go to the DNA. What is the function of the DNA and what is the process and its structure? Okay. Um, So the function of the DNA is as a store genetics information for making proteins. So these involve two processes. So the first one is replication, and the second one is transcription. So the replication, in the process of replication, it synthesizes the DNA into identical copies to pass down to the daughter cells. So DNA as inherited genetic materials. Okay, the second one is this transcription. So the transcription is the process of the RNA synthesis, which is act as an intermediate to make a protein sequence. Okay, and the second function of the DNA is in DNA act as inherited genetic materials. Okay, and it is this is related to its double helix structure. But how? Because of the stability. So because of the deoxyribose of the pentose sugar, it become more less reactive. Okay, meaning that if it less reactive, because of the absence of the OH, and it's more stable as compared to the other one is ribose. Okay, as you can see on your right picture here, on the ribose, the carbon number 2, that's an OH group. Okay, and the other one is deoxyribonuclease. There is no OH, there's only H on it. Okay, and also the double helix, the base pair inside are protected from environmental damage because of the double helix. Okay, so we move on next. The third one, the DNA carries the genetic information, then encode for making protein. So the directionality is 5 prime to 3 prime. This is for replication or transcription. Okay, this will be covered in other topic. In other topic, and there's a base pairing, which is hold the bi-weak hydrogen bond. This is for easiness, for easy to separate the between two strands for the replication and transcription. Okay, and because of the size of the DNA is very long, it can store a lot of information. Okay, and and the packaging is compact, so meaning that when it compacts, a lot of information can be stored in a small space. All right, so this will be covered into the next lecture. Okay, okay, guys. This is the much important. Okay, it's about the central dogma. So, what is the definition of the central dogma? Okay, the central dogma of molecular biology describes the two steps process, which is the transcription and the translation, by which information in genes flow into proteins. Okay, meaning that the process of the transcription and translation can be considered as and not can be considered. It's a process we call a central dogma. Okay, it starts with the DNA and transcription happen and become RNA, and then the translation happen. It will produce a protein. Okay, so in transcription, the DNA sequence of a gene is copied to make a RNA molecule. So this involves rewriting or transcribing the DNA sequence in a similar RNA alphabet. Okay, from the image on your right side here, you can show that it shows that the DNA information storage will undergo the transcription process, and it will produce the mRNA. So mRNA is information carrier, and then this mRNA have a codon. So as you can see here, three different base will code it for one codon. So after that, it translates into the amino acid. Okay, so for codon number 1, UUA, you can see it's a leucine, and then codon number 2 is histidine, and so on and so forth. Okay, in translation, the sequence of the mRNA is decoded to specify the amino acid sequence of polypeptide. Okay, so we move to the next slide. All right, so this is the difference between prokaryote and eukaryote process of the central dogma. Okay, so central dogma in prokaryote is very simple because there is no nucleus to separate the process of the transcription and translation in prokaryote. So bacteria will immediately transcribe, and immediately after the transcription, the bacteria will immediately translate the array. but different in eukaryote, because the eukaryote is quite complex. It's a complex entity. The transcription happen in the nucleus, and after the transcription happen, so the mRNA produced will be transported out to the cytoplasm for the next process. We call it the translational process. Okay. All right. Next, we move to another slide. Okay. So what is a genetic code? Okay, so the genetic code is a set of rules that define how the four-letter code of DNA. The four-letter code of DNA is the TACG. Okay, so this TACG is translated into 20-letter code of amino acid. So from four, it can become it can produce 20 different amino acids. So this amino acid is how DNA sequence is translated into protein sequence. Okay, so what is codon? So codon cells decoded mRNA by reading their nucleotides in a group of three. So in the group of three amino acid, it will become one codon. Okay. So the features of the codon is most codons specify an amino acid. It has three bases or three nucleotides, which represent one codon. There's also a stop codons. There's the three stop codons marked at the end of the protein, which is UAA, UGA, and UAG, and only one start codon, which is AUG, or we call it a methionine. Okay, so this is the table of the genetic code. Okay, so from the four letter that I said before, the DACG, or in this case a UCAG, it will become a 20 different amino acid. Okay, you could see here some of the code some of the amino acid are some of the bases, or some of the codon will translate into the same amino acid. I'm sorry, same amino acid. Okay, so how are codons read? So start codon, let's say for this figure, the start codon is AUG, okay, methionine, okay, it starts from there. 3 AUG, the first 3 is considered start codon, and then the next ACUC is a second codon which encode for the isoleucine. So the isoleucine is AUC, and then the serine UCG, and then it stops at the stop codon, which is the UAA. So this is how the codons read. Okay, so mRNA codons are read from 5' to 3'. All right. Okay, then we move to the reading frame. So from this figure, you can see three different reading frames. So it depends on where it starts. Okay, for the first frame, you can see it starts from the UCA, which is encode for the serine, and then it goes to the UGA. It stops. Okay, that's the stop codon, and then continue again for serine, arginine, and lysines. And frame 2, if you start at the C, so it will become a different amino acid there. So when you start at the C, CAU, it becomes histidine. Okay, and after that GAU, aspartame, and then leucine and valine and arginine. Okay, for frame 3, if you start from A, so it will start the methionine. See, so AUG will become methionine, and then isoleucine, and then serine. Okay, so different frame, different start reading will cause the different frame. Okay, so we go to the RNA types. So RNA is a ribonucleic acid, or it's a change of nucleotide. Okay, so the structures consist of the nitrogenous base, which is AGCU, different with the nitrogenous base in the D and the deoxyribonucleic acid. That the U, the U replaced with the T. Okay, so it also has a sugar ribose, and it's usually single-stranded, but it also has a secondary and tertiary, which is in loop because of the self pairing. Because of its linear, when they meet the G and C together, then it will become a loop. Okay. All right, so there's a three types of RNA. The first one is mRNA, and then tRNA, ribosomal RNA, and there's also others RNA types, which is also known as a microRNA, and the other one is small nuclear RNA. All right, so we start with the mRNA. So mRNA conveys genetic information from DNA to ribosome for protein synthesis. Okay, so this mRNA carries a message from DNA to protein. Okay, in every three nucleotides of RNA, meaning that if three nucleotides is considered as one codon, so it encodes one amino acid. Okay. All right. So in prokaryote, mostly DNA is transcribed to mature mRNA that does not require processing in cytoplasm, but different in eukaryotes, whereby the DNA which transcribed into the mRNA, it will the mRNA is a pre-mRNA, or we call it the unprocessed mRNA. So the pre-mRNA is processed to mature mRNA by the post-transcriptional modification. So by this process, the mRNA produced will be modified to make it more useful, or to make it more to make to cater the protein required. Okay. Now we move to the post-transcriptional modification. So what is the post-transcriptional modification actually? So it's a process where the RNA transcript is first made in eukaryotic cells. Okay, it's considered a pre-mRNA and must be processed into messenger mRNA. So in this process, there's three different types of process happen on the mRNA. So if you have the mRNA from 5' to 3', so on the 5' cap, the 7-methyl guanosine is added at the beginning of the RNA. So this is important to recognize by the ribosome purposes. Okay, and the second one, the 3' polyaethyl will be added at the end of the RNA. So this is very important to protect the pre-mRNA from nucleus degradation. And the last one, in splicing, some sections of the RNA transcript must have an intron, which is the sequence that we don't want, and the other one, so these introns will be removed by the spliceosome, and the remaining sections, which is exon, will be stuck back together. Okay, this is very important. Okay, and then so this will produce a functional protein, as introns are found in the DNA but do not encode it, as it hence need to be removed before protein synthesis. All right. Okay, so this is the schematic of the post-transcriptional modification that I explained before. So the first the first image is a DNA. So in the DNA, there's a promoter and a terminator. So in the middle of it, it has an RNA coding sequence. So once the transcription happens, it will produce the pre-mRNA. So like I said before, the PTM, or the post-transcriptional modification, happen in here. Okay, whereby the 5' cap has been added at the beginning, and the polyethyl has been added at the end of the mRNA. Okay, so in the middle you still have the exon and intron. Okay, remember that exon is the one that you want, and the intron, the exon is the one, the fragment that you want, and the intron is something that you want to remove. Okay, and then from the pre-mRNA, it will process to remove the intron. So this will leave the exon part only, and then after this is a true mRNA, and then after the processing happen, it will translate into the specific protein or polypeptide. Okay, the other PTM process, an alternative for the post-transcriptional modification, is a alternative splicing. Okay, so some genes can be alternatively spliced. So this will leading to the production of different mature mRNA molecules from the same initial transcript. So meaning that if they have one transcript that contain like 4 genes, so maybe it can be spliced into 3 genes or 2 genes. Okay, so alternative splicing is when the exons of the genes are joined differently to produce different protein isoform. So from different exon, from different numbers of exon will produce a different type of protein. So this will synthesize more proteins with different functions, which using a limited set of DNA sequence. Okay, so why do we need the isoforms? So because the isoform is very important to perform more specific functions in different conditions. All right. So this is the post-translation modification types. Okay, the first one is cassette exon, meaning that the first exon can bind to the tree and to another two exons. Okay, and it also can bind only for two exons. So it will produce a different types of protein. And second one is the mutually exclusive exon, meaning that the first exon can be bind either the second exon or the third exon, okay, to produce a different isoform. All right, okay. The third one is intron retention, meaning that in this case the intron is not removed, so it retains as the same in some cases. Okay, and then next is alternative 5 prime splicer site, whereby the second axon can be cut in the middle of it. Okay, and it produce a shorter axons. Okay, and of course it will produce a different types of protein. Okay, and then it can be also spliced at the 3 prime site, and the last one alternate promoters, whereby if there's a 4 axon there, you can find that the promoters might be bind to the second axon. So the transcription will happen at the second axon to translate into the new protein. So now you need to know the different types of RNA, and you can also describe each function. For example, the rRNA, tRNA, mRNA, and hrRNA. Okay. All right. So for this slide, it's just want to show that what is the mature mRNA structures. So the mRNA structure should be in a eukaryote, because no mRNA in the prokaryotes. Okay. And in this coding region, they must have the code. It contains the codons, and the start codon AUG will end up with the stop codon, which is UAA, UAG, and UGA. And also in the mature mRNA structure, there's also the UTR, or we call it the untranslated regions. So these regions is not translated into amino acid sequence. Okay, so the roles of the UTR is for mRNA stability, and also used for the transport and translational efficiency. So there's a two different OUTR, which is 5 prime. So 5 prime is a leader sequence, and the other one is 3 prime, it's a tailor sequence. So the reader sequence start before the codon, and the region for the 3 prime region will start after the stop codon. Okay, then in mRNA also there's a 3 prime poly A tail, and last one is 5 prime cap at the beginning of the mRNA. Okay. Alright, so we go to the ribosomal RNA. So the ribosomal are a small organelle which consists of large subunit and also the small subunit. Okay, as you can see from the figure here, the total weight of the rRNA consists of 60% of rRNA, and the 40% remaining is a protein. Okay, so in eukaryotic ribosomes, the size of the ribosome, the rRNA, is 80S. As compared to prokaryotic ribosome, is only 70S. Okay, meaning that if the 80S is much larger than the 70S. All right. So the ribosome is the site of protein synthesis. This ribosome can catalyze the assembly of the amino acid into polypeptide chain. Alright, there's two different locations for the rRNA. So RNA can be found in the cytosol. So in the cytosol, we call it a free ribosome. Okay, so in a free ribosome in cytosol, it can produce and release protein in cytosol for usage within the cells. But if the rRNA can found in the membrane-bound ribosome, or on the rough endoplasmic reticulum, this is to produce a protein whereby to transport the protein out of the cells. Alright, so this is the difference between prokaryotic and eukaryote ribosome. Okay, in prokaryote Ribosome, the size of the ribosome is 70S, which is have a two subunits, which is 50S and 30S. And then for eukaryotes, the size of the ribosome is 80S, consists of 60S and 40S. Okay, and S is a Svedberg unit. So the Svedberg unit is based on the sedimentation coefficient, meaning that when the ribosome is centrifuged for high-speed centrifuge, it sedimented at different level. Okay, so a different level will make them a different value of the Svedberg. Okay, and meaning that the bigger size is the higher S value. All right, the other one is transfer RNA or tRNA. So this tRNA can carry amino acid to the ribosome, which acts as a bridge that matching the codon in the mRNA with the amino acids it codes for. Okay, so each tRNA contains a set of three nucleotides, which is called anticodon. So anticodon will hold the amino acid that they carry. Okay, the anticodon of given tRNA can bind to one or few specific mRNA codons. Okay, so these codons are complementary with the anticodon sequence. So the anticodon, the part whereby it will bind to the mRNA on the mRNA. Okay, so this is the structures of the tRNA. That, like I said before, the RNA is a linear, right? So if it's a linear, there's also the secondary and the quaternary structures. So this is one of the examples of the quaternary RNA structures. So it become secondary and quaternary because of the binding of the threads. Okay, like you can see here, the C can pass with the G. So when it meet together, it will bind. So it will have a hydrogen bond. So it will become not a linear, but it in a quaternary conformation. Okay, from this tRNA, you can see at the top of it, there's a amino acid attachment site. So this is where the amino acid is bind to. Okay, so the tRNA has also the anticodon site, which is the down part of the tRNA. So this anticodon will bind to the mRNA, which is it depends on what is the mRNA coding for. Okay, all right. And this is the peptide synthesis, where as you can see that there's an mRNA in the middle of the ribosome. So they need a ribosome as a place to put or to carry the tRNA carry the amino acid and then bind to the mRNA, and then it will translate into the protein. So I also prepared a summary of RNA here, so you can see the different mRNA and different RNA between mRNA, RNA, and tRNA in terms of the function, description, and strength. Okay, I think you can look at it on your own. And then I also prepared a summary of the DNA versus RNA. So in terms of the structure, stability, the function, and location. Okay, so I hope that you can explain how the quality and information of genetic materials are conserved in generation after generation. So from the beginning of this lecture, I have told you that the structures of the double helix itself can give conserve the nitrogenous base inside in the middle part, because it's conserved from it cannot be damaged because it in the middle, so it's covered by the phosphate bone at the outer part of the double helix. Okay, and then it also can be transferred from another organism to another organism, meaning that from the mother cell to the daughter cells. I think that's conclude all. Okay, thank you.